Hi, everyone. Welcome to our Tuesday get-togethers on the cerebellum. Good to have you with us, and so pleased to have the honor of introducing David Linden, one of the most eloquent and thoughtful neuroscientists that I know of. David went to UC Berkeley as an undergrad, where he worked with Joe Martinez, um, publishing insightful work even when he was just barely out of high school. Then he went to Northwestern, where he worked with Arya Rudenberg, working on LTP. Then I did a postdoc with John Connor at the Roche Institute, where he worked on calcium imaging, the making of the technique for the calcium imaging. David has been studying cellular basis of memory storage, recovery of function after brain injury, and he is a well-known author who has written three books, Touch, Compass of Pleasure, and Accidental Mind. And being at Hopkins has allowed me to listen to his insightful work, but also listen to him as he asks questions, which are often some of the most insightful questions that you hear. David, it's great to have you. Thank you for taking the time.
Well, thanks, Reza, and thanks to all of you for having me here today. It's a little weird to be speaking at a cerebellum seminar because, as I think you may know, my lab actually hasn't worked on cerebellum for about the last 10 years. We've been doing other things related mostly to axon regeneration in the adult mammalian central nervous system, something that isn't supposed to happen, but does in some cases. But I'm not going to talk about that today. And what I'm going to do is speak in very general terms about plasticity, about use-dependent electrophysiological changes in the cerebellum. And I'm not going to actually show any data slides at all, because all the work that we did on this has been published many, many years ago now, and there's nothing new that we have to contribute. So this might be very brief, but let's go to it. So all right, so glutamate using synapses are found in the cerebellum, the parallel fiber to Purkinje cell synapse, the parallel fiber to interneuron synapse, the climbing fiber to Purkinje cell synapse, the mossy fiber to granule cell synapse. These are all glutamatergic synapses. And all of them have been shown to undergo persistent use-dependent synaptic strengthening, LTP, or weakening, LTD. And we have spent a lot of time on some of these various phenomena. I'll talk for the moment now about the parallel fiber to Purkinje cell synapse, and I apologize for my lousy drawing that kind of looks like I have a baseline tremor, but I really don't. So here we got the presynaptic side. Here we got the postsynaptic side. Has AMPA type ionotropic receptors. Then over here at the edge, it has a different kind of receptor called mGlu1, which is a metabotropic G protein linked receptor that activates the enzyme phospholipase C. And so quite a few years ago, actually, when I was a postdoc in John Connor's lab, following up on a paper by Francis Crapel, we showed that LTD of this glutamatergic parallel fiber to Purkinje cell synapse was triggered by three separate events in the postsynaptic side: ligation of AMPA receptors and activation of mGlu1 and activation of calcium influx through voltage sensitive calcium channels. And we did this in cell culture. It was meant to mimic the climbing fiber parallel fiber conjugation that was originally done by folks like Ito, who discovered this phenomenon. And the idea was that the climbing fiber would produce a massive depolarization that would activate voltage-sensitive calcium channels, producing an enormous calcium influx. The glutamate released from the parallel fibers, particularly when the parallel fibers release in a little burst, rather than when a single action potential invades, would activate both AMP-type glutamate receptors and this G protein link receptor mGlu1, and that these signals pretty much converged on the enzyme protein kinase C, which phosphorylated components in the postsynaptic density that involve the clathrin-dependent removal of AMP receptors from the postsynaptic side. So this form of LTD is acting on ionotropic fast glutamatergic transmission. It is both induced postsynaptically and expressed postsynaptically. It's induced by these three postsynaptic events, and it's expressed by the internalization of postsynaptic AMPA receptors. And so we were all very, very happy about this. And then we were working a little more, and we thought, well, you can't really have LTD at a synapse without either having it return to baseline rather quickly, or preferably having LTP to reverse it, because you don't want to have a situation where your synapses have been LTD'd so much that they're just pinned at their minimal gain. And it turns out that actually you can completely silence a parallel fiber to Purkinje cell synapse. You can LTD it into nothing. You can remove every last AMP receptor from the postsynaptic side if you do it repeatedly, and if you are lucky. I mean, you got to keep in mind that the number of receptors that we're dealing with here is pretty small. We're talking about tens. It's not thousands of AMP receptors in the postsynaptic density. It's tens of receptors. So removing them all is not asking a lot of the cell biology of the synapse. So we went to try to find an LTP, and the LTP that we found, that is produced by repeated burst firing of the parallel fiber alone, is a completely different phenomenon. Rather than being induced and expressed postsynaptically, what we found was a form of LTP that was induced and expressed presynaptically, involved calcium influx through voltage-gated calcium channels into the presynaptic terminal, the activation of the enzyme adenylate cyclase type 1, the activation of protein kinase A, cyclic AMP-dependent protein kinase, and then the phosphorylation of some different substrates in the machinery of vesicle docking and fusion that reduced the probability of release of a vesicle fusing with the presynaptic membrane. So now here is a weird situation, right? So in one sense, you go, oh, okay, all right, you got a synapse, it's got LTP, it's got LTD, maybe it is prepared to participate in dynamic processes and in learning and adaptation. But the problem here is that because the LTD is expressed postsynaptically, whereas the LTP is expressed presynaptically, these phenomena interact, but they actually don't reverse each other, right? In other words, I think it's important to think about what's actually going on at the molecular level here, right? LTD is the removal of AMPA receptors from the postsynaptic membrane, the internalization through clathrin-mediated processes. The LTP that I've described to you is a presynaptic process. That's happening on the other side of the synapses. It is independent of AMP receptors. So what that means is that you could saturate LTD and you could saturate LTP both, and they would be pinning each side of the synapse at its maximum. And there was a period of time where this was kind of what we understood, and we thought, well, that is what we know, but it can't be the whole story. And sure enough, with time, we and others, mostly others in this case, found a presynaptically mediated LTD and a postsynaptically mediated LTP. So both sides of the synapse have both LTD and LTP, which means any one of these processes can be actively reversed. And my own strong opinion is that you need that if you are going to have plastic synapses that participate in learning. So really, what I'm talking about today are not so much phenomena. I mean, first of all, these are all things that are well known and well published. But the point of it is not so much for cellular electrophysiologists like me who've heard and thought about all this stuff. It's for folks working at a somewhat higher level, folks who are thinking about circuit function and who are making models of the cerebellum. What I'm trying to convince you of are a couple of things. One of them is that you can't just represent a synapse as a weight. And the other one is that the molecular details about how these synapses are altered ultimately matter for the way these circuits function in the real world. Now let's talk about the evidence for this. So let's imagine that you've got a synapse and you're recording an EPSP that is evoked. And this is a fast glutamatergic synapse. Let's say it's mediated by AMP receptors. And this EPSP is 1 millivolt in height. And you induce LTP at the synapse. And of course, now it's a bit larger. And let's see if I can draw this properly. All right. Well, that's ugly, but it's supposed to be about the same. It's supposed to be about the same kinetics and about twice as big. And you might say to yourself, well, all right, that's the outcome. Why do I care if this is happening postsynaptically by adding AMP receptors or by changing the unitary conductance of AMP receptors, or presynaptically by some phenomenon like increasing calcium release or increasing the probability of vesicle fusion through interacting with the vesicle fusion machinery? Well, the answer, the reason that you care comes from considering that the physiologically irrelevant signals at this synapse and at many synapses are not single EPSPs, but rather are brief bursts. So parallel fibers often fire a very typical behaviorally salient parallel fiber signal might be, say, 4 action potentials at, about 100 hertz or so. All right, well, so what does that look like? Well, the first EPSP looks like that. By the time the second one arrives, it hasn't come down to baseline, but it potentiates. Maybe the third one will be like this, and the fourth one like that. And that's a very typical parallel fiber 4 times 100 hertz response. Well, now notice that this is a facilitating synapse, that each subsequent EPSP is larger. Why? It's because this synapse has a very low release probability. Release probability at the synapse is in the ballpark of 0.1 or potentially even lower. It's one of the lower release probability synapses that you'll find in the brain at all. And so just to be clear, what does this mean? It means if an action potential invades a parallel fiber terminal or varicosity 10 times, on average, one of those times will you release a vesicle, right? This isn't the way we diagram and normally think of synaptic transmission as being, but this is how a lot of synaptic transmission happens. It is sparse, it is low probability, but what it means is that the aggregate probability of transmission goes up dramatically with a burst. It's a way of, it's one mode of burst to build burst detection. All right, now let's imagine that this synapse, this facilitating synapse, were to undergo a postsynaptically expressed form of LTP. Let's say there are now twice as many AMP receptors in the postsynaptic membrane as there used to be. Well, now what's going to happen? Like the whole thing will, I'm drawing this very badly, but what I'm trying to draw is that the entire thing is scaled up, but that the relative amplitude of the peaks has stayed the same. I've drawn it so poorly, it's hard to see that, but that's what I'm intending, that if you took this dark blue trace and merely doubled its amplitude, it would be the purple trace. And that's what would happen. That's what happened if it were postsynaptically expressed. Now, what happens if instead you're looking at a synapse that is presynaptically expressed? Well, if you look at a single action, a single EPSP, it doesn't matter. But here is roughly what we had before LTP. And now, after LTP, well, so we're potentiating the first one, that's the same. But now, because we have increased the release probability and we've done it presynaptically, the second one isn't going to be bigger, and the third one will be smaller, and the fourth one will be smaller still. Why? Because we are depleting the readily releasable pool of vesicles in the presynaptic side. So the key point here is that the short-term dynamics and the ability to do burst detection are different when you are modulating synaptic strength with presynaptic expression as opposed to with postsynaptic expression. And so this is why, for example, if you're making a model and you are characterizing a synaptic connection as a weight, you are only partially functionally characterizing. Synapses are not just weights, synapses are dynamics. Is it a facilitating synapse? Or is it a depressing synapse? Or is it one of those synapses that is built to neither facilitate nor depress? For example, as Sasha Dulac's lab has shown at the vestibular afferents to vestibular nucleus. So this is a point that I want to make about this. So I think it's worthwhile thinking about how these plastic phenomena interact with different synaptic properties at the basal state. So at the parallel fiber to Purkinje cell synapse, probability of release is about 0.1. At the climbing fiber to Purkinje cell synapse, the probability of release is about 0.99. As a matter of fact, you could even say it's more than 1, because at the parallel fiber synapse, you get monovesicular release when it succeeds. And at the climbing fiber synapse, often you get multivesicular release. That is to say, a single one of the release sites between the climbing fiber, the axon of the inferior olive, and the Purkinje cell can release multiple vesicles of glutamate. Well, if you have a synapse that is nearly at the maximal probability of release, well, you probably not, you can produce LTD of the synapse, but you're almost certainly going to have to do it. You could do it both presynaptically or postsynaptically. If you want to produce LTP of this synapse, you're almost certainly going to have to do it postsynaptically because there isn't any headroom for the probability of release to get any larger. All right. So, when LTP was first discovered by Bliss and Lomo, it was discovered at a fast glutamatergic synapse, the perforant path dentate gyrus granule cell synapse in the dentate gyrus of the hippocampus. And people thought, well, all right, there's just LTP. This is going to be a special thing of the hippocampus because they knew about HM. They knew the hippocampus was important for memory for facts and events. And they said this is going to be a very rare phenomenon, only in certain brain regions that are specifically memory involved. And it's going to be particularly a property of glutamate synapses and of fast glutamatergic transmission. And all those things turned out to be wrong. It turns out that there's both LTP and LTD, that it turns out that most synapses have them, and not just in the brain, but in the spinal cord as well. They are not ubiquitous phenomena, but they're nearly ubiquitous phenomena. And that they are not just present at glutamate synapses, but they are also found at other fast neurotransmitting inhibitory synapses like GABA synapses, and that, as we'll show you, that phenomena are not restricted to fast neurotransmission, but can impact slow neurotransmission and housekeeping functions as well. So in addition to LTP and LTD of fast glutamatergic synapses, as I've described on both the presynaptic side, it turns out that there are both LTP and LTD of fast iPSCs, of fast GABAergic synapses, and I should mention in the spinal cord of glycinergic synapse as well. And that's probably a phenomenon that's going to be found, I think it's found in the brainstem too. So, so we now know, for example, that stellate cell to Purkinje cell and basket cell to Purkinje cell GABA synapses that are fast because they're acting on GABA A receptors that form that are ionotropic, and so as a consequence produce fast iPSCs, that these synapses can also undergo LTP and LTD, counter to what was originally thought by Bliss and Lomo. And then this was a massive surprise that happened in my lab. And I'm going to tell the story of how it happened in the lab because I think it's funny, and I think it also has a little bit of a lesson. So recall, I said that the parallel fiber Purkinje cell synapse, here's the presynaptic side. The postsynaptic side has AMP receptors that are in the middle of the synapse. And then more towards the edge of the synapse is this mGlu1 metabotropic receptor. So what this means is that in order to activate the metabotropic receptor, the glutamate that's released into the synaptic cleft has to diffuse out and achieve significant concentration to activate mGlu1 at this perisynaptic site. And it turns out that single vesicle fusion events don't do this very well. So as a result, mGlu1 is activated by bursts. Now, remember, I told you that mGlu1 is a metabotropic receptor. So it catalyzes this reaction via G protein. It catalyzes the conversion of phosphatidylinositol 4,5-bisphosphate to 1,2-diacylglycerol and 1,4,5-inositol trisphosphate. But there's also a beta gamma subunit of the G protein, and this moves in the plane of the membrane and activates a transient receptor potential type C channel that is a cation channel that lets ions in. So as a consequence of this TRPC channel, if you stimulate the if you voltage clamp the Purkinje cell and stimulate the parallel fiber synapses with bursts, what you'll see is the fast AMP receptors, and then you'll see this tail out here. All right, so here's the tail. Now, if you put on a drug to block the AMP receptors, then you tend to see the tail in isolation. And if you put on a drug to block mGlu1, then the tail goes away. So the tail is an index of the activation of mGlu1 by burst-released glutamate at the parallel fiber synapse. Now, this phenomenon was discovered, I think, in John Garthwaite's lab originally. It's been seen by a lot of folks. Initially, it wasn't known what ion channel was carrying this. I actually published a paper that got it wrong, that said that this was a sodium-calcium exchanger current. That turned out to not be true. It's as I said, it's a TRPC current. And while we were trying to figure out what ion channel was carrying this current, there was a visiting scientist in the lab that I think many of you know, who's going to be speaking here soon, Sang-Jung Kim. And I said to Sang-Jung, I said, we got to figure out what this current is. And he said, well, all right, let's do a current voltage relationship. Let's voltage clamp the Purkinje cell at different postsynaptic potentials and see where we can get this current to reverse. And by doing that, by finding out the reversal potential of the current, that can give you a clue to what kind of ion channel it might be, or to what ion or ions are permeable, right? So for example, if you were to do that experiment with AMP receptors, you get a reversal potential around 0 millivolts, because AMP receptors are permeant to both sodium and potassium ions. They're a little bit more permeant to sodium and potassium, and the weighted average of the two equilibrium potentials comes out to 0. So this is the kind of experiment, for those of you who are not cellular electrophysiologists, that you can do in an afternoon, right? If your slices are good that day, you could get this answer in an afternoon. So I was surprised when I didn't see Sang-Jung for a few days. And he came back and he said, I don't know what I'm doing wrong. He said, every time I get this slow mGlu-mediated current, and then I depolarize the cell past 0 millivolts, when I go back down to negative potentials, it's just gone forever. It's been obliterated. And I said, Sang-Jung, you've just discovered LTD of the metabotropic receptor. This is actually kind of a big deal. And so he spent a long time figuring out, convincing us that that really was the case, and figuring out how it happens. And so there is, in addition to plasticity of fast glutamate, GABA, and even glycine synapses, there can also be LTD of slow synapses, like metabotropic receptors. So, this is signaling, I think the reason, so why do we care? Why is this important? Well, so this kind of ionotropic receptor mediated phenomena is, these are good for information that you need to act on quickly, like moving eye muscles, for example. Metabotropic information is slow, right? It takes tens or hundreds of milliseconds or even seconds to peak, and then it goes away very fast. And this is just the ion current. We don't actually know the kinetics of what's happening to the diacylglycerol and the inositol trisphosphate that are produced. So this is very slow information. And generally speaking, if you look at the brain and you look at neuromodulators like dopamine and serotonin and norepinephrine that either exclusively or predominantly act via G protein coupled slow receptors, the kinds of information that they convey don't tend to be millisecond scale information. They tend to be things like internal state, like, am I hungry? Am I sleepy? Am I horny? Things that change on a seconds to tens of seconds time scale, not things that change on a millisecond time scale. And of course, you will recall that this mGlu1 is famous for LTD of the parallel fiber Purkinje cell synapse. So it turns out that if you produce a depression of mGlu1, now you make it harder, or if you've gotten rid of it, impossible to induce the LTD of the AMP receptor EPSC. So what does that mean? This form of synaptic depression is actually what we call metaplastic, meaning that it is changing the probability of subsequent other forms of plasticity. All right, so let's go back and talk about the climbing fiber synapse. So here's a Purkinje cell. Actually, we can go back to the intro slide. All right, so this is an actual fluorescence micrograph that was made, I believe, by Hiroshi Nishiyama when he is in the lab. And the green are climbing fibers labeled by a fluorescent dye injection in the inferior olive, I believe. And the red is I believe it's calbindin as a Purkinje cell marker. And you can see the Purkinje cell layer here, and you can see the dendrites, and then you can see these railroad tracks that go up and look like they're running on either side of the initial segment of the dendrite, of the proximal dendrite of the Purkinje cell. And what this is it represents that each climbing fiber in the, each Purkinje cell in the mature cerebellum receives one climbing fiber, but this climbing fiber wraps around the Purkinje cell dendrite and makes many synapses. In the human, I believe it makes about 1,000 synapses. And recall that these synapses have an extremely high release probability. And this is why when an olivary neuron fires and an action potential is conveyed to a Purkinje cells via the climbing fiber, that the depolarization that is produced by the climbing fiber is massive, because you are nearly synchronously activating hundreds of very high probability release fast ionotropic glutamatergic synapses. And so if you were to record there, you get, and say you're voltage clamping the Purkinje cell at, well, it turns out you have to voltage clamp at about minus 20 millivolts, because if you voltage clamp at minus 70 millivolts the way you typically would, you get a current that's so big that it breaks the amplifier and you lose voltage control. But even at minus 20 millivolts, you get an absolutely massive inward current. So this is, you know, this can be 5 nanoamps of current, just ginormous. So the glutamate released at the synapse is acting on AMP receptors. So what happens when you put on a antagonist of AMP receptors, like for example the drug NBQX? Well, it turns out that you nearly completely obliterate. So what I've tried to draw here is that in the presence of NBQX, this line is very, very close to flat, but it's not. And if you turn up the gain on your amplifier, what you find is that there is a current here that is maybe 100 picoamps or so, and it is a little slower than the kinetics of this current. And it turns out that this is a very unusual current. This is the current that's produced by a protein called EAAT4, excitatory amino acid transporter 4. Now, normally, at most places in the brain, when glutamate is released, it is removed from the extracellular space by the action of excitatory amino acid transporters in astrocytes, or in the cerebellum in Bergman glial cells, which are the specialized astrocytes of the cerebellum. But it turns out that Purkinje cells have an unusual situation where glutamate is also removed from the synaptic cleft by being taken up through glutamate transporters into the Purkinje neuron itself. And that's what gives rise to this very small, slow current that you see at the climbing fiber Purkinje cell synapse, even in saturating concentrations of NBQX and also other drugs to block kainate receptors. There's a little bit that's left. Now, it turns out that when Ying Shen was in the lab, he showed that this current that represents the uptake of glutamate into the Purkinje cell, and the reason that it's an inward current is because this transporter moves 1 glutamate and 3 sodium in, and it transports a potassium. It also has this complicated mode where when the glutamate is in the middle of the channel, it also fluxes chloride, so it's a little bit complicated. But the end result of this transporter is net positive inward, which is why this is an inward current. And Ying found that this excitatory amino acid transporter current can undergo potentiation after strong depolarization of the Purkinje cell. So why am I telling you this? Well, the functional properties of potentiating the glutamate transporter current are really unclear. It is probably not information storage. It is probably anti-excitotoxic and homeostatic. But the reason that I bring it up is that it's my strong belief that if you look hard enough at any electrophysiological function in a neuron, you'll find that it undergoes long-term use-dependent synaptic plasticity in 9 out of 10 cases. So plasticity is a ubiquitous feature of the electrophysiological properties of neurons. Actually, I'm not going to talk about spillover transmission. And one of the ways that it is important has to do not with synaptic plasticity, but rather with what we call intrinsic plasticity. Now, intrinsic plasticity is the poor cousin of synaptic plasticity. There are probably 100 papers on synaptic plasticity for every one on intrinsic plasticity, but intrinsic plasticity is a very important phenomenon. And what it is it is use-dependent changes in voltage-gated ion channels that will change things like the propagation of synaptic potentials from the dendrite to the soma and the axon initial segment, that will alter the probability or the pattern of spike firing in a postsynaptic cell. And so when Carlos Eisenman was in the lab, he discovered long-term changes in voltage-gated ion function in the cerebellar nuclear neurons. And so what's the classic way to query this? Well, what you do is you've got your recording electrode in a cell. You're in current clamp mode, so you're letting the cell rest if it has a resting potential. And then you inject ramps of depolarizing current to varying degrees. And of course, first you get not much, and then maybe you'll get one spike, and then maybe you'll get a few spikes, and then maybe you'll get a whole bunch of spikes as you inject more and more of this ramp current. And it turns out that this spiking from direct current injection, this so-called Fi curve, is fairly consistent in most neurons. You can do it over and over again, and you will see for a certain amount of current injection the same number of spikes in the same pattern being evoked. But if you induce intrinsic plasticity, now the number of spikes for an amount of current injection changes. Now, you might think to yourself, well, all right, that is a potential mechanism for information storage, but it must be a rather low fidelity one, right? Because, an average neuron in the brain got a dendrite, and your average neuron has about 5,000 synapses that it receives, right? Purkinje cell is unusual. In a human, it's got 150,000 synapses that it receives, but it does more synaptic integration than any other neuron in the brain, but more typical, about 5,000 synapses. You might say, well, all right, let's say you're changing, say, voltage-dependent potassium channels down here at the soma where it meets the axon initial segment. And then what you would do is you would change the probability of initiating an action potential. Let's say you decrease the probability of it. Well, that now what you're doing is you're changing the throughput of this part of the circuit for any input that is impinging anywhere in the dendrite or the soma of this neuron. Now, we didn't talk about it, but generally speaking, LTP and LTD of synaptic transmission happen mostly in a synapse-specific manner. When you get LTP of a synapse, it doesn't tend to spread to all its neighbors in the dendritic tree. It can sometimes spread a little bit, like to one synapse on either side in the dendrite, but it is mostly input specific. Whereas if you envision this form here of intrinsic plasticity, that is going to change the throughput from all the synapses onto a neuron. So as a result, its information storage capacity of this form of plasticity must necessarily be much lower. Well, that would be true if the only place you could induce intrinsic plasticity was here at the axon hillock or at the soma. It turns out that you can actually induce intrinsic plasticity in dendrites, and even you can produce it from synaptic barrage at particular segments of dendrites. So you might change the voltage gate, some voltage gated calcium channel, or I mean, any voltage gated channel in a patch of dendrite at some distal remove. Well, what does that mean? That means that instead of changing the throughput from all 5,000 synapses that impinge on this neuron, now you're changing the throughput from some subset, maybe the synapses that are received in this patch, and then maybe the synapses that are distal to that patch that have where their synaptic currents have to pass through it in order to get to the soma on the axon hillock to influence spike firing. So in addition to synaptic plasticity, we also have intrinsic plasticity, and intrinsic plasticity has the property that it is not merely changing the throughput of the entire array of inputs onto a neuron, but can be more subtle than that. And with time, there's been more and more attention paid to intrinsic plasticity, but I think it's going to be really interesting to consider how intrinsic and synaptic plasticity interact to build computations in circuitry in the cerebellum and in other regions as well. So the last form of plasticity that I'd like to mention is a subset of intrinsic plasticity, and I call it long-term burst mode plasticity. And so what this means is if you've got a neuron and you inject depolarizing current into it, and it normally responds with a burst of action potentials and then maybe very few more or silence afterwards. This would be a strongly bursting neuron. In a use-dependent and persistent intrinsic plastic fashion, it can be altered so that the total number of action potentials evoked by this depolarization, and this might be, let's say, 100 milliseconds long, the number of spikes isn't changed, but their distribution in time is. So now let's just say, I don't know, how many did I draw here? 1, 2, 3, 4, 5. I drew 6 spikes. So instead of having a burst and relax, now you've got a tonic firing neuron where you've got 6 spikes that are happening with more consistent intervals. So you can actually move the mode of firing of a neuron between tonic and burst firing mode in a use-dependent mechanism phenomenon. This was demonstrated by Wei Zhang in my lab many years ago, and she found that it could go in both directions. So a burster can be made tonic firing, and a tonic firer can be made a burster. And this is not surprising because you can do this by changing the function, the conductance, the voltage dependence, the inactivation of various voltage-sensitive ion channels. And in this case, she showed this in the nuclei, in the neurons of the cerebellar nuclei, but actually this can be found all over the brain. And for those of you who are interested in expression profiling, single cell expression profiling as a technique, I think this is an important lesson because this alteration that happens is faster. It can happen in a few seconds. It's not happening on the time scale of changes in the transcription of genes and the subsequent translation of mRNA. So what it means is that if you are trying to relate firing properties to intrinsic firing properties of a neuron to gene expression profiles, in some cases, you can get fooled. That is to say, it's very likely that the very same neuron with the very same signature of single cell expression profile can give you two different firing modes just based on its particular plastic state when you happened to retrieve it. And so, all right, well, so what are the lessons from all this? Well, if you look carefully enough, most electrophysiological functions of neurons, and I should say, we're just looking into this, but very likely the electrophysiological functions of glial cells, and glial cells, while they're not spiking, have neurotransmitter receptors, both ionotropic and metabotropic, as well as transporters and other interesting things. All of these phenomena are modulated by experience, and the molecular details of synaptic and non-synaptic plasticity matter if you wish to understand the transformation of neural signals by experience and the function of neural circuits. So, basically what I'm saying to those of you who are working at the circuit level and who are building models, don't forget about the biology. The actual details of how things change here turn out to be crucially important for how and what the circuits can compute. So, this whole big block of names is a bunch of people who've been in my lab over the years. You probably recognize a lot of them. Many of them have gone on to have their own labs. Many of them still work in cerebellum, like Christian Hansel and Wei and Ying and Hiroshi and Sunjung. So I put these images up here in case you've been wondering, but what my lab's been up to in the time since we left the cerebellum. The image, the false color image on the left is blood vessels in the living mouse brain. They've been depth coded with the warm colors at the surface and the cool colors showing the depth. And so we've been very interested in the function of the brain's vascular system. And on the right is a section of mouse neocortex. It's layer 1 at the top, layer 2, 3 at the bottom. The cyan blobs are nuclei, and the green axons are serotonin axons, and the red axons are norepinephrine axons. And we spent a lot of time understanding how these axons can regrow after injury in the adult brain, which is something they're not supposed to do. I'd be remiss if I didn't mention many of my wonderful collaborators over the years that have contributed to the kind of work that I've alluded to. And thank you very much for your attention.
Thank you, David. That was wonderful. We lost the image of your face when you moved some things around. I wonder if you could bring that back.
Yeah, okay, maybe is that back?
Yeah, your image is back. Wonderful. So if anybody has questions, please just go ahead and raise your hand, and we'd be happy to. I'll convey it. So, David, you mentioned 10 years ago you stopped working on the cerebellum, and I wonder if you can give us some thoughts about that decision-making process.
Well, so I mean, part of it was just at that point, I started working on cerebellum as a postdoc. And so I had been, it had been over 20 years of working on the cerebellum at a, mostly at a cellular and molecular level. And, I think there were two things. One of them is I just wanted a different challenge. And the other thing is that I kind of felt like a lot of the areas where I could contribute the best had become somewhat more mature, and the areas where my lab could do things that were that were different than other labs was diminishing. The other thing is that, quite honestly, I became so much more encouraged by the state of the cerebellar field. I mean, when I started in 1990, honestly, it was a snake pit. And there was a lot of very ill feeling. There were a lot of opinionated, mean people. And I thought, what did I get myself into? And I saw that it was an area where the level of behavior was so bad that it was keeping many young investigators from starting in cerebellum. It was scaring a lot of people off. And over the years, the personalities that contributed to that have moved on. And as a result, the field, really, I think, became much more exciting and interesting and multipolar and rejuvenated starting about 15 years ago. And so I kind of felt like, you know what, I can leave this field in good hands. There are so many terrific people here that they're not going to it's really not going to be impacted by the loss of me in particular. Also, I got really interested in axon regeneration.
That's wonderful. Okay, Sasha, you have your hand up.
Yeah, David, that was really beautiful. Thank you for reminding me of all of those different forms of plasticity. And I know that you thought that possibly LTD would be the site of the engram. And I just want to know, in the ensuing decades, what's your view now on whether neuroscientists are going to be able to understand anything? Are there engrams? Is this something worthwhile?
Yeah, that's something that I think about, and I even get teased about it when I watch the NFL, because there's a tight end for Jacksonville, and his last name is actually Engram. And so I'm seeing this player on my TV, and it says Engram on the back of his uniform. And I think, yeah, the universe is trying to tell me something there. Yeah, when I started out, we had high hopes that if we could understand LTD at the parallel fiber Purkinje cell synapse in enough detail, that we could produce a specific enough disruption to LTD that it would be a good test of the hypothesis that LTD would underlie certain forms of motor learning, like associative eyelid conditioning or gain up VOR. And so we worked very, very hard to do that. And the problem here that we were fighting against is that, there's a whole tradition of people, say, for example, making mutant mice and saying, oh, this mutant mouse doesn't have LTP or LTD, and it's deficient in this behavior, therefore this form of plasticity is necessary for this behavior. But it turns out it's devilishly hard to do because of divergent signaling. So you guys can still see my desktop, right?
Yes.
Okay. So, for example, if you were to look at LTP in the hippocampus, right, you would say, oh, all right, well, you got NMDA receptors, and they influx calcium, and that activates CAMK2, and then CAMK2 phosphorylates some stuff, and you have a substrate, and then that gives you LTP. And so people said, we'll put on NMDA receptor antagonists and see if we can block LTP in the hippocampus, and we'll see if it'll interfere with behavior. And sure enough, you put NMDA receptor antagonists in the hippocampus, and you block things like, Morris water maze learning. And you go, oh, all right, but it turns out that the NMDA receptor fluxes calcium. Calcium isn't just there to activate CAMK2, it's very broadly signaling. So this isn't really much of a test. How do we know that blocking NMDA receptors has its effect on the behavior from blocking LTP versus any one of the other things it does. And so he said, oh, all right, well, we'll go down instead, we'll interfere with CAMK2. Well, but the problem is CAMK2 has hundreds of substrates in neurons. And so it also phosphorylates stuff. So you have the very same problem. Even inhibiting CAMK2 will not allow you to conclude that the loss of LTP produced by CAMK2 inhibition underlies a change in some behavior that you measure. So we thought, all right, well, we got to understand this in the case of cerebellar LTD. We've got to understand this enough to get to the substrate, right? And so we found out that the GLU-A2 AMPA receptor is phosphorylated by PKC at serine 880, and so it turns out that you can not just mutate this serine, but mutate a nearby one. You can turn a lysine number 882 to an alpha that doesn't make it unphosphorylatable, but makes it unphosphorylatable only by PKC. And we thought this is a really specific mutation, right? This probably isn't doing that much other than interfering with this form of LTD. This will actually be a good test. And so, we did this together with Chris Dzeu's lab and John Oberdick, and also with Rick Hugenier's lab. And sure enough, if it turns out if you block mGluRs, you can block LTD and you can block associative eyelid conditioning. And if you block PKC, you can do it. But when you actually get down to this very specific mutation, that you don't block associative eyelid conditioning and you don't block gain up in the VOR, and Chris has since claimed, well, you need that together with also with one of these. Oops, sorry, I pressed the wrong button. With one of these other phenomena, that is to say, plasticity and the throughput through the molecular layer interneurons, and that may well be true. But I think, the challenge, Sasha, is that even if you can get down to a few places, it's not going to be stored in one place forever, and it's not going to be stored only in one place. It's almost certain to be stored in many places in a circuit, and that is going to change over time. So I would say, I'm not going to say that relating that sort of finding engrams, so to speak, is something that is ultimately impossible. But I think it's going to be a whole lot more difficult than I had imagined it 25 years ago. The other thing that I would like to say, just because you mentioned the word engram, is that these days engram is used in a way that I don't like very much. People do mini scopes and they see neurons that are activated like during memory retrieval, and they say, well, these are engram cells, and to me that's a bit of a bridge too far. That is not proof that those cells actually are participating in the storage of that information. That is a mere correlation. So anyway, that's my little rant about the word engram.
It was lovely. Are there any other questions for David?
Hi, David, if I may, thank you. Can you say more about this plasticity at this the GABA synapses? Is it driven by climbing fiber input? Can you say more about the ideas of spillover to molecular layer to these stellate or basket cells?
Oh, all right. Well, so okay. So there's a couple of different phenomena here. So as you probably know, the climbing fiber makes direct synapses. If you look in electron micrograph, onto the cerebellar Purkinje cell, but the glutamate that spills out of that synapse can then, through pure spillover, activate glutamate receptors on molecular layer interneurons. And it turns out that that phenomenon, that if you do, for example, LTD of the climbing fiber Purkinje cell synapse, you will also depress, you can also depress the spillover transmission if you do it in some ways. So I think the whole idea that there is pure spillover transmission is really exciting and interesting, and is not something that people have talked about very much. In terms of the plasticity of inhibitory synapses, this is not, my lab worked a bit on the Purkinje cell to cerebellar nucleus synapse plasticity. There's been a whole lot of work done, starting by, I think, Masanobu Kano and Arthur Connerth years ago, on the molecular layer internon to Purkinje cell synapse. And my understanding is, my recollection of that, it's been a long time since I read these papers, but that form of LTP of those fast GABAergic iPSCs was postsynaptically expressed as GABA A receptor function, and that it could indeed be triggered by the climbing fiber or by artificial depolarization of the Purkinje cell to mimic the climbing fiber.
Thanks.
Thank you, everyone. It's wonderful to have you. David, thank you so much for taking the time. This was a wonderful tutorial. Certainly was very, very informative for me.
All right. Well, thank you for inviting me, even though I've been out of the fold for so long. I still love the cerebellum, and I certainly enjoy being in the audience at these seminars regularly.
Thank you so much, David. Have a wonderful Tuesday.
Bye-bye, folks.